going to start by So start by thanking the person who really did the work, who's sitting back there, Paul Umbra. Everything I'm going to present is basically her PhD project, and if you have, I'll try and do it justice. If you have questions, the person to ask is sitting back there. Stephen, thanks also to our collaborators, Stephen, who gave us a lot of advice along the way. Eric Massé, who we worked with on the microbiome part of what I'll show you. Mariano Avino, a bioinformatician who helped us with the bioinformatics on the studies. And this work was basically performed here at the Health Sciences campus of the University of Sherbrooke, and our lab is in the Pavilion for Personalized Health and Translational Medicine, the PSPR. I'll have the talk is divided in 5 sections: background, experimental approach, the results concerning the brain, the results concerning the body, and then a little take-home message. So, Alzheimer's disease, I won't spend much time on this. We all know that the cases of dementia worldwide are ballooning by 2030. 2030, yeah, about 75 million people expected with dementia. The main cause of dementia being Alzheimer's disease, and in Canada, the incidence of Alzheimer's disease is about 7% in the population, and of course, a big age-dependent increase. So by age of 85, almost 1 in 4 with Alzheimer's disease. When we think about Alzheimer's disease, we think about the brain with reason, learning and memory problems, anxiety, depression, etc. One of the things that's becoming clear is that there are also peripheral consequences of Alzheimer's disease, which consequences are maybe involved in the pathogenesis of the disease. So there's emerging data and convincing data and emerging data on links with the diabetes, with obesity, hypercholesterolemia, and other peripheral conditions. Another idea that's coming out, and which is getting more and more support, is that before we have the late onset of cognitive symptoms in Alzheimer's disease, there are many risk factors that are taking place earlier during life. So see early life education, midlife, cholesterol, hearing loss, depression, traumatic brain injury, physical inactivity, diabetes, smoking, hypertension, obesity, excessive alcohol, etc. So many things that we do prior to reaching the age of onset of Alzheimer's disease can affect the development of Alzheimer's. And up to 45% of this risk is thought to be now potentially modifiable risk factors. And so if you look at what's happening during the pre-symptomatic period of Alzheimer's disease, some of the processes that are involved are amyloid deposition in the brain, tau pathology, but we see also changes in metabolic activity in the brain. And this has, of course, led to things that you've heard about already. So the idea of restoring brain metabolic health in Alzheimer's. So with FDG PET, we see that in Alzheimer's disease, there's reductions in glucose uptake in the brain, glucose being the main fuel, of course, and that's probably contributing to the generalized atrophy of the brain seen in Alzheimer's disease. And this has, of course, led to the ideas championed by Stephen and other people about rescuing brain metabolism as a therapeutic strategy. So we've already seen this slide, the reduction in cerebral metabolic rate with age, and the idea of trying to restore that by improving the contribution of ketones to brain energy metabolism. And so in the context of mild cognitive impairment or Alzheimer's disease, there's evidence that this approach can work, either using exogenous ketones, as we've heard them called today, or endogenous ketogenic interventions. And so there is evidence in the literature, in particular from Stephen's lab, showing that these approaches can be beneficial in the context of mild cognitive impairment and potentially Alzheimer's disease. And some of the nicest work has come from Stephen's lab, and there's people in this room who have done this, and it's an honor to be associated with your work. So effects on with medium chain triglycerides, effects on cognition, white matter energy supply, processing speed, functional and structural connectivity, for example. And so, do we have the answer to Alzheimer's disease? And of course, the answer is no. So, even there are now systematic reviews that have come out looking globally across the literature, and basically what these show is that there are mixed results in the literature. Some studies showing positive results, some studies showing not so convincing or negative results. And even within studies where there are positive results, you tend to see these types of things where there are non-responders. So people that increase ketones and yet don't have the improved cognition. And so one of the questions is, why is there such variability in the responses to ketogenic interventions? And so the research objective for this particular project was to help try and clarify the cellular and physiological mechanisms underlying the encouraging effects of ketogenic interventions on cognitive function. The experimental approach that we're using is to use preclinical mouse models of Alzheimer's disease. And I'll explain that in a moment. Basically, we're using two different animal models of Alzheimer's disease. These are transgenic mice which express human Alzheimer's disease-inducing mutations. And we're using two particular models. One which has a relatively light phenotype, and so it's particularly useful for modeling early stages of Alzheimer's disease. And one, the 5X-FAD model, which is a much more aggressive model, and so is useful for modeling late stages of Alzheimer's disease. Now, there's different ways of using clinical mouse models to gain useful information. So one approach is called forward translation. And the idea is, if you use preclinical mouse models for discovery research, because there's reduced noise from genetic variation and from different life experiences, so you can get very reproducible results. And then to take the findings that you have and translate them towards human to see if they work. And so this approach is useful for identifying cell biological basis of development, disease, and aging processes. These are processes that are conserved across species. There are also attempts to identify novel therapeutic targets in mice and apply them to humans. And so this works pretty well. This, as we know, doesn't work as well because mice are not humans, and a lot of things work in mice that don't work in humans. But there's also a reverse translation approach. And so this is where you take treatments that are already showing promising or encouraging results in humans, and you model them in mice to try and understand how they're working and optimize how they're working. So we would do this to try and optimize existing interventions, and also by understanding how they work, to have a rational design of combinatorial strategies that harness different molecular mechanisms. And the idea is that because you're starting with an intervention that's already showing promising results, there will be a higher forward translation potential at the end. So this is the approach that we were using here. And so we used two different mouse models, as I mentioned, a 3xTG model with a very slow pathogenesis process, which allows us to model earlier model events in the disease, and the 5X-FAD model, which is a very aggressive model. And so this is just an example. So if you look at wild type or the Alzheimer model, and you see the background staging just to show the hippocampus structure, what you see is that at 2 months of age, the Alzheimer mice are relatively normal. And by 5 months of age, you have the development of all these plaques that are characteristic also in the human Alzheimer's disease. Okay, so we used these animal models using different ketogenic interventions. So a control condition with 70% carbs, 20% fat, 10% protein. An MCT condition in which half of the fats were medium chain triglycerides, and a ketogenic diet in which the carbs were completely removed and replaced with fats. So the theory is that in the MCT condition, where you have a high level of carbs, a small amount of MCT, you'll have the body will be running mainly on carbs and ketones generated from the MCT. In the ketogenic diet, where the carbs are replaced by the fatty acids, now you'll be generating large amounts of ketones to power the body. Okay, so we generated these diets, we fed them to mice for different periods of time. Do they work as expected? So the mice were put in metabolic cages, which allow you to measure the respiratory exchange ratio, and tells you whether the body is running mainly on lipids or on carbohydrates. So when the respiratory exchange ratio is closer to 1, it's running mostly on carbohydrates. When it's closer to 0.7, it's running mostly on lipids. And you can see that in the wild-type mice and in the Alzheimer mice on the ketogenic diets, okay, it's close to 0.7, as you would expect. Control mice on the control diet and on the MCT diet, the mice are running more on carbs. So this is basically as expected. And if you look at the same thing over a 24-hour period, so you see that the dark cycle, so the active cycle in mice, because mice are what's the term? Nocturnal, thank you. So you see both the wild-type mice and the Alzheimer mice on a ketogenic diet are running all the time on lipids, as expected. And you have the nocturnal increase in activity and decrease in metabolism that is basically unchanged by the presence of the MCT diet. We measure actually the ketone levels in the blood. What you see is that in the wild-type mice, the green is the control diet, the blue is the MCT diet, and basically you see there's no increase in blood BHB. On the ketogenic diet, you have the expected large increase in BHB levels. And something similar in the Alzheimer mice, although you see the rise in the ketones, it's not as much. So before starting the long-term experiments, this is what we had. So when we looked at this data, basically the expectation was, well, maybe the MCT diet is not really going to do very much because the ketones were not increased. The circulating ketone levels are increased by the KD, but not by the MCT supplementation in these mice. So then we began the analysis of the brain. And so here you see the brain of a mouse. Okay. And you see in particular the green, which is the hippocampus, which is the region of the brain involved in learning and memory, and which is also a region that degenerates and atrophies very significantly in Alzheimer's disease. So that's the region that we focused on for these analyses. And so we used one of the models of the Alzheimer models. We raised the mice to 6 months of age and then put them for 1 month on these diets. And in these animals, by 5 months of age, the brain is already full of plaques. So this is a late stage intervention. After 1 month on the diet, we then did behavioral testing for learning and memory. And we did that using the Morris water maze. So the way the Morris water maze works is you have a pool and you have a water that's usually got milk powder to make it slightly opaque, and you have a platform in the pool. And you put the mice in the pool, and they don't like to be in the water, so they try and find the platform. And over several days, you train them in the pool. Basically, they're only point de repère, or only guideposts are these signs on the wall. So it's testing their spatial memory and their spatial learning ability. So there's 2 phases to the test. When you train them every day, you see very rapidly over 4 days, they learn where the platform is. And so differences in how quickly they find the platform and an approximation of changes in their learning ability, spatial learning ability. Now, on the 5th day, test their memory as opposed to their learning, you take away the platform. And you see how much time they spend in the quadrant looking for where the platform used to be versus in an opposite quadrant. And so those are the two parameters tested in this test. During the spatial learning phase, what you can see is that their latency to find the platform, so the number of seconds it takes for them to find the platform, the wild type mice, over 4 days, they rapidly learn where the platform is. This is basically unchanged by the MCT or the ketogenic diet. The wild type, the Alzheimer mice, what you can see is they fail to have the same learning curve. So their learning ability is reduced. But what you can see is that when they're placed on the MCT or on the ketogenic diet, basically their spatial learning ability is restored. When we look at the memory phase, we see again something different. So here you see the time in the target quadrant where the platform used to be versus the opposite quadrant. You can see there's a significant difference, significantly higher in the target quadrant than in the opposite quadrant. And this is true in the MCT and in the ketogenic diet also. When you look at the Alzheimer mice, there's no longer a significant difference between time in the target quadrant and opposite quadrant. But when they're on the MCT diet or on the ketogenic diet, that preference was restored. So here you see the 5x5 mice, so the Alzheimer mice. In the control diet, there's no real preference for where they're spending in the pool. On the MCT diet, and on the ketogenic diet, they're spending more time where the platform used to be. So both the MCT and the ketogenic diet improve spatial learning and spatial memory in the Alzheimer mice. One of the advantages of using preclinical modeling, now we can look in more detail at what's happening in the brain. And so we looked at hippocampal dendritic spines. Are the ketogenic diets, ketogenic interventions affecting the structure of the brain? And so with this approach, what we do is Golgi staining. And here you see a section of the brain stained with Golgi. And here you see neurons with the plastic. Okay. So here you see different types of neurons within the hippocampus with their long dendrites. And if you zoom in on the long dendrites, what you're able to see is these dendritic spines. In Alzheimer's disease, these dendritic spines start to disappear, and that correlates with learning and memory problems. So if you zoom in on these dendrites, what you see is this. And here you can see the dendritic spines along the dendrites, and you can quantify the density of those dendritic spines. When you do that, what you see is that both in the wild type mice and in the Alzheimer mice, the density of dendritic spines increases on the MCT and ketogenic diets. It's about a 20 or 30% increase in dendritic spines. So the structure of the synapses is changing in these mice. So both MCT and ketogenic diet increase the density of dendritic spines on hippocampal neurons. And we went even deeper. So we looked at gene expression. So I isolated the hippocampus of wild-type mice and of Alzheimer mice on the 3 different diets, isolated the RNA, and examined the expression of their genes, all 20,000 genes within the genome. What we observed was there were about 600 differentially expressed genes in the Alzheimer brain. So significant proportion of genes that were dysregulated. When you looked at what pathways they were involved in, what you could see is that in the Alzheimer mice, there was increases in mitochondrial energy metabolism genes, decreases in synaptic function genes. So as you might expect. What happened to the mice on the different diets? So of these 600 genes, 41% of them in the MCT diet were no longer significantly changed. On the ketogenic diet, 57% of them were no longer significantly changed. And of those, 34% were rescued by both diets. So at the genetic level, these diets are restoring the normal gene expression patterns in the hippocampus. And the effects of the 2 diets are shared, but not identical. So at the level of the brain, both MCT and ketogenic diet improves learning and memory, increase dendritic spines, and reverse the gene expression changes in the hippocampus. But recall, MCT is doing this without increasing the levels of circulating ketones. So how is it doing this? So this led us to examine what are these diets doing to the rest of the body? Could they be affecting other systems that are then having the effects on cognition? And so, as I said earlier, Alzheimer's is increasingly associated with changes in peripheral systems in the body. When we look at the Alzheimer mice, which are carrying mutations in the brain that cause Alzheimer's disease, we also see peripheral changes occurring in these mice. So, for example, the spleen is significantly enlarged in these Alzheimer mice. Different fat stores are increased: abdominal fat, subcutaneous fat, brown fat. Blood glucose is also increased in these mice. So we asked whether could we use these mice to begin looking at what is changing in the periphery in response to these diets. So where do we look in the periphery? So if you think about the way diets are ultimately affecting cognition, so they have to pass through the digestive system, where they can potentially affect the microbiome or the enterocytes of the gut. They then pass through the portal vein, through the liver, into the systemic circulation. From the systemic circulation, they can affect many different tissues. So a lot of indirect effects possible on what's changing in the systemic circulation. Eventually, they'll reach the vasculature of the brain, where they have to pass through the blood-brain barrier, enter into the parenchyma, and then have the effects on brain cells and alter cognitive function. So a long road. The diets could be affecting any of these things. So we've already seen that they're restoring brain cells and cognitive function. So we looked earlier on this pathway. We asked, what about peripheral metabolism? So we looked at some general markers of peripheral metabolism. So using glucose tolerance and insulin tolerance tests and changes in body weight. So here we have another model of the Alzheimer model. They were placed on diets for 6 months, and we tested them for these different parameters. This is an example of the glucose tolerance test. So similar to in humans, if you inject glucose, you examine the rise and how quickly it goes back down. And so this is the wild type mice on the control diet, the Alzheimer mice on the control diet. See, the Alzheimer mice have a hyperglycemia. The Alzheimer mice on the MCT diet, not too much different. The Alzheimer mice on the ketogenic diet, you see, if anything, the opposite effect of the MCT. The insulin tolerant test shows, again, something similar. Injection of insulin, blood glucose goes down. With the Alzheimer mice, they are, again, hyperglycemic compared to the wild type mice. The Alzheimer mice on the MCT become almost like the wild type mice on their insulin tolerance test. And those on the ketogenic diet seem to be having the opposite effect. We look at body weight gain per caloric intake. The Alzheimer mice and the wild type mice on the control diet, the Alzheimer mice are gaining more weight per caloric intake than the wild type mice. On the MCT diet, there's now no longer any significant difference. And on the ketogenic diet, the Alzheimer mice are gaining even more weight compared to the wild type mice. So in the Alzheimer mice, the MCT supplementation and ketogenic diet seem to have opposing effects on insulin, glucose sensitivity, and on body weight regulation. So it's probably not by that these two diets are having similar effects on the brain. So we looked earlier in the chain, and we asked, what about the liver? We take sections through the liver, even just with histological staining. You can see the Alzheimer liver by H&E staining, by lipid staining using oil red O, or by using ODIPI, visually they look different. So we looked in more detail, again, doing RNA sequencing. So we took the liver, similar as we did to the brain, we took the liver of wild-type mice or Alzheimer mice on three different diets. Compared wild-type Alzheimer mice on the control diets, there were about 843 genes that were significantly downregulated in the Alzheimer mice or significantly upregulated in the Alzheimer mice. So big changes at the genetic level in the liver of these Alzheimer mice. Ones that were upregulated were lipid synthesis genes, pathways. Many of the ones that were downregulated were lipid oxidation genes. So what happened on the diets? 44% of the differentially expressed genes in the Alzheimer mice were rescued in the liver by the MCT, 55% by the KD, and again, 30% rescued by both. Similar to the brain, big effects on the gene expression in the liver, shared pathways, but ketogenic and the MCT pathways on diets, but not identical. So the AD mutations induce significant alterations in the liver that are largely corrected by MCT and KD intervention. So the liver seems to be the place where these diets are potentially acting. And then Paul decided to look at the microbiome. Okay, so here what you see is the two models, the early and the late model, and RNA sequencing of the feces. Tell you basically here what we're looking at is the taxa of the different microbial species in the feces. These are volcano plots that show taxa that are significantly upregulated in red or increased, taxa that are significantly downregulated in the Alzheimer mice in blue. And you can see there isn't not exactly the same taxa in the two different models, probably because they're different stages of the disease that are being modeled, but significant numbers of microbial species that are altered, upregulated or downregulated in the Alzheimer mice. So what is the effect of so the AD models have baseline changes in the gut microbiome. What is the effect of the diets? So here you see one of the AD models. You see this, what I just showed you, the upregulated and downregulated taxa, the upregulated and downregulated taxa in their feces. And here what you see is the AD mice on the MCT and the AD mice on the ketogenic diet. And in green are all the rescued taxa. So taxa that were upregulated or downregulated and that are now no longer significantly different. So the MCT supplementation and the ketogenic diet both largely restore the taxa dysregulated in the AD mice. We look in a little more detail. So now each mouse has hundreds of different taxa. If we want to visualize this, you use in a simpler way, you use dimensionality reduction. And so this is what my team has become very good at, bioinformatics. But you use dimensionality reduction. So what you see here is projected in three-dimensional space, each mouse that was used in the studies. And what you would see if you could read the labels was that the wild-type mice are all here on the control diet. The Alzheimer mice are all there on the control diet. On the MCT diets, you see they shift. So there's a remodeling of their microbiome. On the ketogenic diets, they shift even more. So the microbiome is remodeled even more. So MCT and KD both remodel the gut, but the gut microbiome, but differently. If we ask what is it actually doing to the microbiome, there's different indexes of microbiome diversity. What you see here is that the wild-type mice on the control diet versus the Alzheimer mice on the control diet, the microbiome diversity of the Alzheimer mice is reduced. And what you see here is that the Alzheimer mice on the MCT and on the ketogenic diet, the microbiome diversity is coming back towards normal. And that is generally considered a positive for health. So both MCT and KD restore microbiome diversity. So to summarize all that, what we can say in these animal models, that the MCT supplementation and the ketogenic diet had similar effects on the brains of baby mice. You're looking at learning and memory, synapses, and gene expression. At MCT did this without increasing circulating ketone levels. The MCT and KD had opposing effects on systemic energy metabolism, glucose regulation, insulin sensitivity, and body weight regulation. But the Alzheimer mice also exhibited liver and gut microbiome disturbances. And these were significantly reversed by both MCT supplementation and KD. So it's raising the question of whether these diets are affecting other systems in order to have their effects on the brain. It raises the question, are the gut microbiome and the liver potential mediators of the effects of ketogenic interventions on the brain? And so does this make sense in what we know from humans so far? So there are studies showing association between liver alterations and Alzheimer's status. And there's now many different studies, so it's the systemic review of those studies, showing that there's also evidence for the gut microbiome being dysregulated in Alzheimer's disease. So those two systems seem to be dysregulated in Alzheimer's disease. Potentially the diets are acting on those to improve cognition. And so Paul looked, last panel here, so Paul looked in her data to see whether she could draw any correlations between changes happening in either in gut microbial species or in changes in gene expression within the liver, and correlate that with, on a per animal basis, with the number of synapses in the brain. And there are some interesting correlations that are there. This is an example of just one of the genes in the liver that is modulated by the diets, showing a very strong correlation with the numbers of dendritic spines in the hippocampus. So that's work that will be built on in the future to try and understand whether there are specific pathways in the liver or microbial species in the gut that are potentially candidate pathways or microbial species affecting the brain. So with that, I'll end there. Thank you to members of the team and our different collaborators and funding agencies, and any students are looking for places. But thanks.